What's the penguins? Today we do 2018 number 4. It's on gene expression error bar analysis. So the common bedbug is a species of insect that is becoming increasingly resistant to insecticides. Bedbugs possess several different genes that are contributing to that resistance. We have P450, ABC8, and CPS. And so the investigators want to do is investigate the role that each of those is going to have in the insecticide. So they deleted one or more of these different genes in these different strains. So here we see in strain 1 that it has all 3 of the different genes. Strain 2 is missing P450, strain 3 is missing ABC8, strain 4 is missing CPS, and strain 5 is missing both P450 and ABC8. So the first question is identify the control strain in the experiment. So I'm able to say that my control strain is going to be species 1. I know this is my control because I'm investigating how each of these genes is affecting the survivability of these different bedbugs. And so I'm comparing without each of the different genes to my control, which would be my species 1, because it has all 3 of those different genes in there. So the second question says, using the means and confidence intervals of figure 1, justify the claim that ABC8 is effective at providing resistance to this certain insecticide. So if I look here, I can see the strains 1, 2, and 4 all have ABC8, while strains 3 and 5 do not. So if I'm comparing the different strains, I can see the strain 1 and strain 3 both have P450 and CPS, and the only difference between these is just looking at ABC8. And so if I look at those error bars, because this prop told me to look at the error bar, I can see that the error bars between 1 and 3 do not overlap, which means that this data is statistically significant. I see that without having ABC8, I'm seeing that it is going to have lower survival, and that the error bars between my control and strain 3 do not overlap. And so this possible answers you could have put, error bars or confidence interval in strain 1, control, wild type, do not overlap with strain 3 or the ABC8 deletion. You could say that the mean percent survival of strain 3 falls outside that 95% confidence interval of that strain 1. So strain 3, or the one that has the ABC8 deletion, shows a statistically significant difference from strain 1 or our control. So the student says strain 1 was the control strain. ABC8 is effective at providing resistance because when ABC8 was deleted on 20% survived, and the confidence interval between strain without ABC8-3 and the control did not overlap, meaning there's a reliable statistical difference between having ABC8 gene and not having it. So B says that we're looking at the P450 encodes an enzyme that detoxifies insecticides. ABC8 encodes a transport protein that pumps insecticides out of the cells, and CPS encodes an external protein that reduces the absorption of the insecticides. So based on that information, I need to explain how a deletion of both P450 and ABC8 results in lower survival when I compare to just CPS alone. So if I look at deleting the 450 and the ABC8, that's looking at strain 5. And I can see that it does have a lower amount of survivability. Like it causes most of the bed bugs to die. And so we want to think, well, why is this taking place? Well, P450 encoded something that would detoxify the insecticides. So if the insecticides got into the cell, it was able to detoxify and get rid of those insecticides in a way and make them not effective. And then also ABC8 encodes a transport protein, and that would pump the different insecticides out of the cell. And so this strain is unable to detoxify, and it's unable to transport those insecticides back out. The only thing it's able to do is just reduce the absorption of the insecticides. But those insecticides that do get in are going to cause it to die, which is what we have here with strain 4. We're looking at the deletion of P450 and ABC8, and it only has CPS. So bed bugs can neither detoxify nor pump out the insecticide, which results in a lower chance of bed bug survival. So if I look instead at strain 4, I have both P450 and ABC8, but it's missing the CPS. And you can see that it does have a higher amount of survivability, okay? Because this one is going to be able to detoxify and is able to pump the insecticides out. Yeah, maybe it can't reduce the amount that are absorbed, but once it does get into the cell, it's going to be able to detoxify and pump back out, which is what the answer the scoring guidelines say. So if they're present, CPS being absent, the bed bugs can detoxify and pump, which results in a higher chance of bed bug survival. So the student says when both ABC8 and P450 are deleted, the cell cannot get rid of the insecticide that has been absorbed. So because CPS doesn't encode for an exoskeleton that completely stops insecticide from getting in the cell, any insecticide that goes into the cell remains toxic and has no way of leaving. When just CPS is deleted, the survival rate is much higher because although there's a higher concentration of insecticide is getting into the cell, is getting detoxified and being removed from the transport proteins encoded by ABC8. So I hope that was helpful. Remember, AP Bio Penguins is just success. Bye.